Hey, Mr. Martin's IB biology class. We're tackling proteins with you today.
That's right. Big topic.
Huge. Looks like Mr. Martin sent us everything but the kitchen sink to help break it down. Notes, textbook chapters, even some study guides snuck in there.
He wants to make sure you guys really get this, and for good reason.
Yeah, proteins are.
Yeah.
Well, they're kind of a big deal.
Kind of. They're involved in almost every single process in living things.
Okay, yeah. A big deal.
What I find amazing is how much variety you get from just 20 building blocks.
Okay, so that's what I don't get. We're talking about amino acids, right?
You got it.
So how can just 20 amino acids create all the different proteins?
Think of it this way. Imagine you have a necklace, right?
Okay.
And you've got 20 different beads to choose from. You can make a short necklace, a long one. You could even make one that's super complicated with branches and loops.
All using the same 20 beads.
Exactly.
Okay, I see that. But there's got to be more to it than just the order of the amino acid.
Oh, absolutely. Each amino acid has a unique structure.
And that structure dictates how it interacts with the other amino acids, right?
Exactly. So let's break it down. Each amino acid, it has a central carbon atom.
Like the anchor?
Yeah, something like that. Then you have an amine group,
Okay.
a carboxyl group, a hydrogen atom.
And the mysterious R group.
There it is.
I remember this from class.
The R group is where things get really interesting. That's what makes each of the 20 amino acids unique.
So some R groups are attracted to water.
Right. Those are the hydrophilic ones.
And some repel water. Hydrophobic.
Exactly. And some even carry electrical charges.
It's a molecular party.
I like that. And all that variety is what gives proteins their incredible range of properties.
But how do those amino acids actually link up? I got peptide bond in my notes here somewhere.
Okay, so picture two amino acids coming together.
Okay.
One loses a hydrogen atom from its aminamine group. The other loses a hydroxyl group from its carboxyl group.
And then they join together.
And that lost hydrogen and hydroxyl, they pair up too. They form a water molecule.
Wait, so water is like a byproduct of this whole process.
That's right. It's called a condensation reaction.
And the bond that forms between those two amino acids is the peptide bond.
Exactly.
Okay, and two amino acids linked together make a dipeptide.
Right.
And you keep adding more.
And you get a polypeptide, a whole chain of amino acids.
Which then folds into some crazy three-dimensional shape to become a functional protein.
Exactly. And that folding process, that's crucial.
So it's driven by those R group interactions.
Right. Plus, a little help from hydrogen bonding along the backbone.
Okay, so now we've got our amino acid chain all linked up. But Mr. Martin also mentioned that humans can only actually produce 9 of the 20 amino acids.
That's right.
So what about the other 11?
Well, those are what we call essential amino acids.
Essential because...
Because we have to get them from our diet.
Oh, okay, so like vitamins.
Exactly. Your body needs them but can't make them itself.
So if we don't eat a balanced diet and get all our essential amino acids...
Your body can't build the proteins it needs.
That's a pretty big deal.
It is. Especially for anyone following a vegetarian or vegan diet, they need to be extra mindful of getting those essential amino acids from plant sources.
Makes sense. So let's say we've got all our amino acids then, essential and non-essential. How does the body know what order to put them in to build a specific protein?
That's where DNA comes in. It's the blueprint for protein construction.
Our genes,
which are segments of DNA.
Contain the instructions for the precise order of amino acids.
Right.
So each gene codes for a specific protein.
Exactly.
That's a lot of information packed into our DNA.
It is. And it all comes back to that amazing diversity of proteins we were talking about earlier.
Right.
Even a tiny change in the amino acid sequence, maybe just one different amino acid, can completely change a protein's shape and function.
So with just 20 amino acids, the possibilities are basically endless.
It's more than just the number of amino acids. Think of it like this. Each amino acid is a letter in an alphabet. With that, you can create countless words, sentences, and even whole books.
Just by arranging the letters differently.
And by changing the length of the sentences.
The length of the chains.
Exactly. That's the power of protein diversity.
Okay, I'm starting to get the bigger picture.
Good. And remember, each protein has a unique shape.
Right.
And that shape determines its function.
So we're talking about proteins that are tiny, massive, soluble, insoluble.
You got it. The possibilities are incredible.
Mr. Martin put some examples in his notes.
Oh, yeah. Do any of them ring a bell?
Well, I remember learning about insulin. That's a pretty small protein, right?
It is, relatively speaking. And it helps regulate blood sugar levels.
Right.
Insulin's a great example of a protein hormone. It's perfectly designed to bind to receptors on cells and signal them to take up glucose from the blood.
It's like a tiny key that fits into a lock.
Perfect analogy. And then you have things like enzymes.
Which are like little molecular machines that speed up chemical reactions.
Exactly. They're a bit more complex than insulin, structurally speaking.
So different structures for different jobs. Makes sense.
But there's a catch. Protein structure is pretty sensitive. It can be affected by things like temperature and pH.
Like cooking an egg.
Exactly. The heat causes the proteins to unfold and clump together. That's what changes the texture.
Ah, denaturation. I remember that word.
So when a protein loses its shape, it can't do its job anymore.
Game over.
Pretty much. Denaturation can happen from heat, changes in pH, or even exposure to certain chemicals.
Okay, this is all starting to make sense. We've got our amino acids. They link up to form chains. The chains fold into shapes, and then they become functional proteins. Wait, there's still more, right? I remember Mr. Martin talking about different levels of protein structure.
You're right, we've only just scratched the surface. Are you ready to dive deeper into the fascinating world of protein folding?
Let's do it. Okay, so different levels of protein structure. I'm ready. Let's unpack this. Primary, secondary, tertiary, and quaternary structure.
It's all about building on the basics.
All right, like we already talked about the primary structure.
Exactly.
That's just the sequence of amino acids, like the order of beads on a string.
It's determined by the DNA sequence of the gene.
And it determines everything that comes next.
Right.
Okay, so secondary structure, what happens there?
So this is where things start to take shape, literally. Sections of the polypeptide chain, they start to coil or fold.
Into predictable patterns.
Exactly, because of hydrogen bonding.
Between the amino acid.
Between the backbones of the amino acids.
Hold on, backbones.
The backbone is like it's the repeating sequence of atoms in the polypeptide chain.
Okay.
Except for the R groups.
So it's like the spine of the protein.
Exactly.
Okay, got it. So the hydrogen bonds form between those backbone atoms.
And that causes the chain to twist and turn.
Into those specific shapes.
Yep. And there are two main types of secondary structures.
Alpha helix and beta pleated sheet.
You got it.
I think I saw diagrams of those in Mr. Martin's notes. What do they actually look like?
Okay, so imagine an alpha helix, like a spiral staircase.
Okay, I can see that.
It's coiling tightly around a central axis.
Hmm.
And the hydrogen bonds, they hold the turns of the staircase together.
Got it. And what about beta pleated sheets?
So picture this. A sheet of paper, but folded back and forth like a fan.
Okay.
That's basically a beta pleated sheet. Hydrogen bonds between adjacent strands hold those pleats together.
So you get that flat sheet-like structure.
Exactly.
So we got coils and sheets. Where do we go from there? What happens next?
All right, well, things get even more interesting at the tertiary structure level. This is where the entire polypeptide chain folds into a complex three-dimensional shape.
And this is all driven by those R groups.
Exactly. So those R groups, they interact with each other.
And with the water around them.
Right.
Some are hydrophilic, some are hydrophobic, and some have charges.
That leads to a whole variety of interactions. All those interactions help fold the protein into its final form.
It's like all those R groups are trying to find their perfect spot, either hanging out near the water.
Or hiding from it.
Exactly.
For example, the hydrophobic R groups. They tend to cluster together on the inside of the protein, away from the water.
While the hydrophilic ones stay on the outside.
Yeah, they love to interact with that aqueous environment.
Just like a molecular dance party.
Yeah.
With everyone trying to find their perfect partner and position.
I like that. So in addition to those hydrophobic and hydrophilic interactions, you've also got things like ionic bonds.
Between charged R groups.
Right. And then there's disulfide bridges. Those are strong covalent bonds that form between certain amino acids.
So much going on. All right, so we've gone from a straight chain to coils and sheets to this complex 3D shape. Is that the final level?
For some proteins, yes. But for others, there's one more level.
Hit me with it.
Quaternary structure. This is for proteins that are made up of multiple polypeptide chains.
So instead of one chain folding up on itself,
You have multiple chains all interacting with each other. Each individual chain has its own tertiary structure. But then they all come together in a specific arrangement to form a functional protein complex.
okay.
Think of it like this. A team of specialized workers all coming together to complete a really complex project.
Do you have any examples
Yeah.
of proteins with quaternary structure?
Yeah, absolutely. Hemoglobin.
Hemoglobin. That's the one that carries oxygen in our blood.
Exactly. It's made up of 4 polypeptide chains. 2 alpha chains and 2 beta chains, all linked together.
So each chain can bind to oxygen.
Right.
And then together, they form this super efficient oxygen carrier.
Exactly. The way those chains are arranged within the hemoglobin molecule, that's crucial. It lets it pick up oxygen in the lungs.
And then release it where it's needed.
You got it. Okay, so we've got primary structure, secondary structure, tertiary structure.
And sometimes quaternary structure.
Right.
That's a lot of folding. But I still don't get, how does this folding process actually happen? Is there something that guides the protein, makes sure it folds the right way?
That's a great question. And honestly, it's one that's puzzled scientists for decades. For a long time, people thought that proteins folded spontaneously. Like the amino acid sequence had all the information the protein needed.
This is like a built-in GPS system.
Something like that.
Mhm.
But the reality is, it's a bit more complicated. The amino acid sequence is a big factor, but there are other forces at play too.
Like what?
Well, there are these amazing molecules called chaperone proteins.
Chaperone proteins.
They help guide the folding process. They can actually prevent misfolding or aggregation.
Aggregation.
That's when proteins clump together and can't function properly.
So chaperone proteins are like the protein folding police, making sure everyone behaves and ends up in the right spot.
Haha, I like that analogy. And there's growing evidence that the cellular environment itself plays a role too.
Really?
It's crowded inside a cell, all those other molecules bumping around. It can affect how a protein folds.
Wow, it's like a delicate dance.
It is. The amino acid sequence, chaperone proteins, the cellular environment, all working together. They make sure that proteins fold correctly.
And protein folding isn't just a one-time event.
Oh, definitely not. Proteins are constantly being made, broken down, and refolded in our cells. It's a super dynamic process and makes sure our bodies have the right proteins in the right shape at the right time to do all the jobs they need to do.
This is amazing. I had no idea protein folding was so intricate
Yeah.
and so dynamic. But what happens if a protein doesn't fold right?
That's a really important question because when proteins misfold, they can lose their function or even gain toxic properties.
Wait, toxic. How could a misfolded protein be toxic?
Well, think about it this way. A misfolded protein, it's like a piece of machinery but with a broken part. It can't do its job, and it might even jam up the whole system.
Yike.
Sometimes misfolded proteins can even aggregate, forming clumps that interfere with cellular processes. They can even trigger cell death.
So misfolded proteins are like little saboteurs, wreaking havoc in our cells.
Good analogy. And there's actually a growing body of evidence linking misfolded proteins to lots of diseases.
Like what?
Alzheimer's disease, Parkinson's disease, even some types of cancer.
So what can we do about it?
Well, that's the million-dollar question, isn't it? Scientists are working really hard to try and find ways to prevent misfolding, promote correct folding, or even just clear out those toxic aggregates from cells.
Sounds like there's a lot more to learn.
Definitely. The world of protein folding is full of challenges and opportunities. But before we go too far down that rabbit hole, maybe let's take a step back, review what we've learned so far about protein structure.
Okay, good idea. Quick recap of those 4 levels of protein structure.
Sounds good to me.
Okay, quick recap time. We started with the primary structure. That's the sequence of amino acids.
Just a straight chain.
Then secondary structure,
Where hydrogen bonding between backbone atoms kicks in.
creating those alpha helices and the beta-pleated sheets.
Right, and then we've got the tertiary structure, where the entire polypeptide chain folds into a complex 3D shape.
Driven by all those R-group interactions. And sometimes proteins even have a quaternary structure.
When multiple polypeptide chains team up.
And we can't forget about those chaperone proteins, helping guide the folding process, preventing mishaps.
Which can lead to some pretty big problems.
Yeah, we covered that.
So to really get all of this, let's connect those structural concepts to some actual proteins and their functions.
Okay, good idea.
Remember those examples Mr. Martin mentioned in his notes?
Well, there was collagen, which I always think of as like the scaffolding that holds our bodies together.
Exactly. Collagen, it's a fibrous protein,
Right.
and its strength comes from its structure.
That triple helix.
Yeah. Imagine 3 polypeptide chains all wound up together like a really strong rope.
So that's why collagen is so important for skin elasticity, tendons, ligaments.
It's felt tough.
And then there was hemoglobin.
Ah, hemoglobin. That's the one responsible for carrying oxygen in our red blood cells.
Which has that quaternary structure.
You got it.
It's a bit more complex.
Hemoglobin is made up of 4 polypeptide chains. Each chain has a heme group that can bind to oxygen. It's a great example of how well, how the arrangement of those subunits, it creates this super specialized function.
Like a team of oxygen carriers,
Yeah,
all working together.
exactly.
Oh, and what about enzymes?
Enzymes, another diverse group of proteins.
Mostly globular, right?
Right, and they act as catalysts. Their shapes are crucial for that.
It's all about those active sites, where the enzyme can bind to its specific substrate and then speed up the reaction.
Like a lock and key. Only the right substrate can fit into that enzyme's active site.
Perfect analogy.
And, without enzymes, a lot of the chemical reactions in our bodies would just happen way too slowly to support life.
So enzymes are important for everything, from digestion to DNA replication.
Pretty much.
It's amazing how these little molecules have such huge effects.
Then don't forget about antibodies.
Oh, right, antibodies.
Another key group of proteins, part of our immune system.
They're the ones that fight off pathogens, right?
Yep. Antibodies. They recognize and bind to specific antigens, like the ones found on bacteria and viruses.
And they have that Y shape.
That's important. Two arms of the Y. They contain the variable regions.
The variable regions can bind to lots of different antigens
Right. And the stem of the Y, that part interacts with other cells in the immune system,
to trigger a whole cascade of events.
which helps neutralize the threat. So each antibody, it's like a weapon, customized to target a specific enemy.
That's so cool.
It all comes back to those 20 amino acids and how they fold and interact. It creates this incredible diversity, different shapes, different functions. It's remarkable.
This has been an amazing deep dive. I didn't realize how much complexity
Yeah.
and how much elegance there is in the world of proteins.
And we've only just scratched the surface, really.
There's so much more to learn about protein folding and how they interact.
And their roles in health and disease.
Mr. Martin's class. If you take away one thing from all of this, remember this. Proteins are the workhorses of life. Understanding how they work, their structure, their function. It's key to understanding so much. How our bodies work, how diseases develop. It can even help us develop new therapies and technologies for a healthier future.
Keep asking questions, keep exploring. Who knows? Maybe one of you will be the one to make the next big breakthrough in protein research.
And on that note, our protein deep dive has come to an end. Thanks for joining us.